Okay, so let's just round out the last parts of this, and these are things that would only be on as like an extra credit, and where we're not going to need to know the entire pathway, because that's more a graduate level immunology thing, I would say. So, additional proteins that do something for you in the way that RIG1 does something for you, or TLRs, or NOD-like receptor proteins. Another one of those is going to be a protein called STING. And STING, S-T-I-N-G, which of course stands for a big long acronym, but we don't need to know it here. We're not going to be STING experts. This can actually bind to double-stranded DNA. And so, yeah, there's going to be a whole pathway there. It's going to involve making interferon response factor number 3, which goes to the nucleus and starts to make interferon alpha and beta, the type 1 interferons. So, yeah, the only thing you want from this is that there is the STING pathway, and the STING pathway, yeah, leads to interferon, and it's activated by double-stranded DNA. Another one of these pathways that is worth considering later on when we're talking about dendritic cells and the way that they function. So dendritic cells and macrophages have CD14 on the outside. And I think you're going to need to make up some mnemonics, MNEMONICS. These are just ways to help you remember the different proteins and what they do, because all of these things have something like a code name. But just like you know 007 is James Bond, you can figure out that CD14 is the thing that is on a dendritic cell or macrophage and will help it bind to bacteria in a way that activates the macrophage. So that's going to be the trick, is coming up with your own, I don't know, some little rhyme or something you can connect it to. And you're building up practice in this with the complement pathway, with the toll-like receptors and their adapters and their localization, whether they're on the surface or interior, and their cell type, at least one out of the nice long list. Yeah, and that's that's a good skill to have. That's a good skill to build. So we're building that as much as immunology here. But yeah, if you're interested in how these things work, this is really important. When you get a cut in your skin and there are little bacteria in there, this is how we make a response, and this is how you don't die every time you get a little tiny scrape wound or every time you cut yourself shaving or something like that. So you've got these skin-resident dendritic cells, that's this yellow guy up here in the top, and they make a couple of these genes. So one of those is going to be TLR4, toll-like receptor 4. Do you remember what toll-like receptor 4 binds? I bet you do. If not, go back and look it up on your little chart, and then you'll have it. Yeah. So reinforcing, that's what we want to do. So TLR4 can bind something that's on the surface of bacteria. Maybe even a component of the cell wall. I don't know. But we'll find out, or you'll know. Yeah. And then CD14 is going to bind to other components. And so with CD14 and TLR4 binding to bacteria, this is now enough of a signal that the dendritic cell is going to pull the bacterium in, digest it, and race, run, not walk, to the nearest lymph node. And once it's there, it's going to show little pieces of the bacterium it just ate to the rest of the immune system. And we'll see if we can make an adaptive immune response there. But yeah, these markers are important for all kinds of cells with phagocytic activities. So cells that eat other bacteria, debris, cells, whatever. Yeah. If you're curious, this is not something I'm going to test on, but if you're curious, the actual toll protein in fruit flies works this way, and it does have little tear domains, TIRs, at the bottom, so it looks very much like our toll-like receptors. Hence, that's why they're toll-like receptors. But instead of, you remember that one of the toll-like receptors, I believe it was TLR4, had one of these little proteins that sits inside of it and modifies it. Well, in the fly, they have a protein called spatzel, and that one gets cleaved, and that will actually sit in the center here and modify it, cause these two to join together. And then once they are bound together, and a number of things can set off this cleavage cascade that activates spatzel, then you're going to have a similar sort of signaling. Your tier domains are going to go through, and we're going to have different protein names at the end, but they're basically the same proteins. We're just looking at different copies of them that have evolved in a fly instead of the copy that's evolved in us. So you remember which proteins were in this position before? It was the Iraq proteins. Yeah, you remember those? Iraq 1, Iraq 4? Yeah, fine. And and so then instead of binding onto the, pardon me, E2 and E3 ligands, or ligases, sorry, which are going to add ubiquitin to things, they're going to bind to cactus kinase, which is directly going to phosphorylate cactus, which is then going to break down. And you notice how cactus looks like IKK, and you notice how DIF looks like NF kappa B. These are proteins that kind of look the same and are doing the same job. Everything that's alive is kind of built of the same parts, just being reused in slightly different configurations, and that is very much the case here. So the pathway that keeps a fruit fly from getting fungus on it is kind of the same pathway that's helping you recognize bacteria that are in your skin or elsewhere in the body. So now let's continue on. So, all right, we're done with our generic pattern recognition receptors, and now let's talk cytokines and chemokines and their receptors, because these things are going to be involved. I believe all the cytokines and chemokines, at least the ones that I know of, are going to go and signal through G protein coupled receptors. And then some of these things are going to be associated with JAK kinases, which are Janus kinases. JAKs are going to activate STATs, and this is involved in a number of different signaling mechanisms, so like type 1 and type 2 interferon signaling, which is stuff we're going to pick up down the road. So let's get an overview of this sort of stuff, and we'll pick up the things that we really need to learn later on. But for right now, let's just go, let's go for the overview. Okay. So, cytokines bind to a number of different proteins. Yeah, sorry, I was mistaken. A number of different receptors shown up here. The chemokines, those are the ones that bind to G protein coupled receptors. So a G protein coupled receptor will normally be shown as an orange snake that goes through the membrane 7 times, just like that. And these are the ones that are hooked up to the G proteins, remember, where it kicks that G protein and it breaks into 2 halves, and the 2 halves go off and do amazing things. And so this is just a little overview of ways to bind different cytokines and chemokines. And there are also things like tumor necrosis factor alpha that's not called technically a cytokine or chemokine, but it's effectively doing the same thing. It's just a different version of this. Okay, so cytokine receptors, contain at least 2 chains. And the 2 chains are going to come together, and each chain basically does hold on to half of the cytokine. So the 2 chains are only together when there's a cytokine bound to them. And each of these chains has a thing called a Janus kinase, or JAK, that is associated with it. And the Janus kinase will then activate each other. So one will put a phosphate on here, and the other will put a phosphate on here. And so then once our Janus kinases are activated and have their phosphates on, they will then transfer these phosphates to a protein called a STAT. So this is JAK, is Janus, Janus kinase. So these are named after, well, I guess you pronounce it Janus, who's a two-faced god. You see him on doorways a lot in ancient Rome. And so you can see it coming and going, basically. And so the idea of these JAK kinases is that it takes 2 of them working together, and they'll each phosphorylate each other. That's actually pretty common. You have a lot of kinases that work that way. But it may also be from, they can have different effects. Some of these effects will be interferon promoting, some will be interferon reducing, basically. All right, so the JAKs are going to transfer these phosphates to the STAT proteins. And STAT proteins are signal transduction and transcription proteins, if I remember correctly. And so these STAT proteins are then going to go to the nucleus once they're phosphorylated, so they'll act like an IRF3, or an IRF7, or an NF-kappa B. When these things get phosphorylated, they're going to go to the nucleus. Or in the case of NF-kappa B, when you take off the I-kappa B and phosphorylate that so it goes off and gets destroyed, then it can go into the nucleus. And the only reason these things go into the nucleus is to bind to a specific part of the DNA that's just before whatever genes you need to make to respond to that particular cytokine. So each cytokine will phosphorylate different patterns of JAKs. It'll have different JAKs associated that will phosphorylate different stats, and these stats will then go to the nucleus and bind different but overlapping sets of genes, and cause different responses from the cell. That's the idea. So here are some chemokines that are going to be released by macrophages and dendritic cells. And they are going to bring other immune cells into the site of infection. So this is kind of neat. So remember IL-1 beta over here, and we mentioned that along with IL-18. IL stands for interleukin. Leuco means white, so we're essentially talking about white blood cells. Interleukin are the secret sort of internal communications of the white blood cells. So this is a message that a white blood cell sends to a different kind of white blood cell that causes it to do something. So in general, these are some of the things. Interleukin-1 beta is really strongly pro-inflammatory, and so this is going to drive fever and production of interleukin-6, which is going to drive more fever. So interleukin-6 is something that gets turned on very strongly in COVID-19, and it also gets turned on in some kinds of arthritis. So blocking interleukin-6 or blocking interleukin-1 beta, these are things that we sometimes will do as a therapy targeting the immune system. That's the idea. Okay, there are different kinds of chemokines, and they break down into these different groups. So they are CC, CXC, and CXXXC, or CX3C. And these guys are, so what this means, this CC, CXC, or CXXXC, is that they're going to have two cysteine amino acids with the abbreviation C. And they're either going to be right next to each other, boom, like that, or they're going to have one amino acid, it doesn't matter what amino acid coming in between, so that's CXC, or they're going to have three random amino acids coming between the two cysteines. Otherwise, these are just very small proteins that fold up in a certain way, and there are lots of functions. These are not things that I'm going to make you learn right here, but if you want to know how a neutrophil or a naive T cell gets activated, it's going to be some of these CXC chemokines like interleukin 8. And you'll see also over here CXC L1 chemokine is going to activate some of these young T cells, naive T cells. And then more activated T cells and natural killer cells are going to be driven more by these CC chemokines. And so you can call them by different names. There's like an old-fashioned name, which is like MIP1 alpha, MIP1 beta, or you can call them CC chemokine like number 3, 4, 2, something like that. CCL, yeah. So that's just a little how things work part here. And also, we're still talking basic sort of innate immune system function. You're going to have proteins like selectins. And selectins are going to bind carbohydrates in a similar way to how a lectin would also bind carbohydrates and then be hooked up to something else. So these are little modules that can be attached to other proteins, or they can bring additional functions to the immune system. I would say that's the idea here. So integrins, like this alpha-beta integrin, are going to be very important. These are factors that can call in other white blood cells and cause them to leave the bloodstream and go into a tissue, basically invade a tissue. And so, you'll find integrins on several other kinds of cells, but including the ones that are going to be involved, like white blood cells. And so they're going to bind to proteins that are coming out of the endothelium. And those are going to be things like ICAM1. So ICAM1 is going to be one of the things we put out. and then the integrin on our white blood cells is going to catch the ICAM1, and it's going to be brought into the cell. Or rather, it's going to cause that cell to move out of the vein or artery and into the tissue where it can actually do some damage. So, for example, you got your ICAM1, your ICAM2, and you've got various kinds of integrin. And integrins are always going to have an alpha and a beta subunit, but there are different types of alpha and beta. So alpha M or alpha L, and beta 2 is being shown right here, but there are different kinds of beta subtypes. And yeah, that's what a neutrophil is going to have on it. And so this is another layer of complexity. Depending on exactly which signal the cells of the endothelium, which remember is this is like the wall of your vein or artery, depending on which kind of markers it puts out, it can bring in different kinds of cells. So these would be ways to bring in neutrophils, and there will be other signals you could put out that would cause other white blood cells, like a monocyte to stop and become a macrophage, or other kinds of other kinds of cells, like a T cell, for example, or a B cell. All right, and so these selectins are going to bind to our molecules, where a lectin usually binds to some other sugar, like on a bacterium or something like that. Selectins select the correct immune cell and cause it to enter into the tissue, just like that. They're going to snag it and cause it to come in and be activated, and then boom, yeah. And there's our interleukin 8 here. This is one of the things that naive T cells will respond to. And so, yeah, cells like this will actually just roll along, and neutrophils will also respond to this. They'll just sort of roll along the wall, and when they run into one of these ICAM molecules, that will cause them to leave the vasculature and enter the tissue and see what's there to see what's there to fight. Yeah, these are pugnacious little cells. They like picking a fight with whoever is making you sick. So, this can work if you get a little cut. You can end up with inflammation in just a little part of your body, and that's fine. You can have your little neutrophils invading that part of the body, and there will be a little bit of leakage, which is going to lead to the swelling and some of the pain. There will be a lot of inflammatory mediators, different compounds that are released by these neutrophils and macrophages. And those are going to cause some of the redness and also some of the pain and some of the destruction of tissue in the area. And then as the last result, we're going to have our neutrophils explode and trap some of the bacteria. And we're going to start the coagulation cascade to kind of seal off the area so that we can clean it up and then eventually reopen it. And so that can happen with a localized infection, or you can have it with a systemic infection. Now, this is very dangerous. This is a condition called sepsis, and it's going to be caused typically by gram-negative bacteria. So bacteria where the cell wall is hidden between two layers. And when you rupture those things, if you're doing something or taking a medicine that causes those to break apart, then the middle layer on those is full of lipopolysaccharide, which is going to activate one of those toll-like receptors. And you can go back to your chart if you want to figure out which one. And then what happens there, it can be very bad. So this can cause full-body sepsis, which is a full-on immune response in all of your blood vessels at the same time, which can cause a lot of damage and a lot of fluid to just flow out in organs and all over the body. Buildup of fluid outside the vasculature is called edema. This is when you get tissue with a little pool of, watery stuff that used to be in the blood, basically plasma building up there. And too much edema, and your lungs don't work, and your heart doesn't work, and your liver doesn't work, and you die. This is a good way to end up with multi-system failure, basically, caused by your immune system. So yeah, usually this is what you want. You want the localized version of inflammation. You want to avoid the this sort of thing. And so having a chemokine like TNF alpha, which is tumor necrosis factor alpha, released, Normally, its job is to just sort of keep our immune response reined in. With diseases like severe bird flu or severe COVID or severe Ebola, you do get macrophages releasing TNF alpha all over the place in the bloodstream, which causes this cascade that just leads to more activation, more immune cells attacking various tissues, followed by edema, followed by multiple organ failure, followed by death. So, yeah, severe diseases tend to follow this pathway, and mild diseases tend to follow this pathway pretty generally. And so you can have a lot of different causes, a lot of different viruses or bacteria that will cause TNF to be set off everywhere inside the body. But this is going to be the outcome that's most common, is that it's going to lead to death. And so it's one mechanism, but many ways to set off that mechanism. I think that's the idea. So let's pause here. That's enough for this section. Then we'll come back and finish out this chapter in the next section.